Okay. Yeah. So thank you very much for this opportunity to be here. It's really a great workshop. I like a lot the friendly atmosphere and the level of talks is amazing. So I just wanted to start by flashing a few projects. I won't be able to talk about everything, but I just wanted to mention some of them. So maybe what's the common factor in all this project is that is really growth, and I'm interested in growth for the same reason that Simone Pigolotti explained yesterday, namely that really growth is the hallmark in some sense of biological systems, and there's lots of interesting non-equilibrium statistical physics that go with that. And so we've played with growing system for a while. So here, basically, what we were interested in is in how a system can grow even when the environment is uncertain. What I mean by that is that the environment can be, for instance, fluctuating. And we ask what is a good strategy of for a population to deal with that. And so this is related to models of bed hedging. I think there will be a talk later on that. And so we've done essentially a mathematical model to explain bed hedging. So maybe if this is if you know about this, so we study, for instance, a gambling model called the Kelly model that is helpful to understand this bed hedging strategies. And from this gambling, we got an interest about what is the risk of a gambler, and we wonder what implication this could have in biological systems. So I'll come back to this idea of risk later in the talk. A second topic is a topic of autocatalytic systems, because they are a way by which a system can grow. And here the idea is really to forget about the dynamics, the kinetics of that. Try to think about how a network can grow, and we borrow ideas from economy, because in fact a growing economy satisfies certain constraints which are similar to the ones we have in the metabolism, for instance, in terms of what are the input and output fluxes, for instance. And so these two projects, I won't talk too much in details. I chose to talk about bacterial growth inhibited by antibiotics. It's really a simple model. It's minimal. And I'm sorry, there's no extreme value statistics, but there's not even fluctuations. So you might be disappointed, but I'll try to motivate that this might be a useful model to go beyond to models that might have the features you like. So if there's time, I'll also talk about cell lineage statistics, which I think is more relevant for the conference that we are here. Okay, so how can we understand inhibition of bacterial growth by antibiotics? So for me, I really, I mean, of course, it's a very old topic, and there's lots of things to say about it. But I was particularly interested in this work that came out 10 years ago, because for me it was kind of like the perfect paper in a way, because it combines good simple theory with good data also, and it provides a simple picture of what might be going on when you have an antibiotics which initially is outside the cell, then enters the cell and binds to ribosomes. And what these people have done is that they've used growth laws, which are recent and very important result in the field of cell growth. And together with that, they essentially made interesting predictions about what could come out, and they have found that, for instance, two different classes of behaviors for the growth versus antibiotic concentration. So I had two comments about this. I think it's beautiful work, but I was wondering, for instance, whether you could derive this growth law within the model instead of having them as kind of ad hoc external constraints. And so this is what motivated us. And there was also the issue of whether this scenario that of antibiotics targeting ribosomes can be extended to other types of antibiotics. So in order to address this question, we need a simple model of metabolism, and so for that we relied on more recent work, where basically these authors have proposed a kind of toy model of metabolism, which is based on the idea that there are certain autocatalytic cycles in metabolism. One of them, for instance, is the cycle of RNA polymerase, namely this process by which the cell makes more RNA polymerase. And this one is coupled to also a cycle for ribosomes. So there are not that many cycles that are really essential, but these two in particular look like they are. And so I mentioned that some of the intuition that we have on metabolism come from economics. So I like to emphasize the work of Leontiev, who got the Nobel Prize in economics for introducing the idea of input-output matrices. And so what he did is that he analyzed the American economy with these matrices, and he understood, for instance, the interdependencies of different sectors in the economy. So I think these ideas are potentially useful for metabolism because you can understand interdependencies of different factors in the metabolism from that. Another useful idea that was used in the work I mentioned is this idea of production function, which I summarized with this kind of bike equation. So if you want to produce a bike with certain rate, you cannot do that at a rate which is larger than the minimum of the production rate of the saddle and half the production rate of the wheel. So it's very basic, but you'll see this can guide us towards understanding interesting behavior. So with these tools in hand, so we use essentially this idea of two coupled autocatalytic cycles, and then we combine the Grölich idea, in which basically there's antibiotics that come from outside the cell and then bind to an essential molecule in the cycle. And then once you do that, basically you can focus on one cycle because the amount of these molecules will be limiting, and so this simplifies a lot the description. So to get simple results, we assume a balanced growth. That means that the dilution rate that comes from cell division is balanced by the growth rate of essentially all species inside the cell, and then this can be calculated explicitly. As you'll see in the next slide, and we find the growth rate that is smaller than the growth rate that you have to start with when there's no antibiotics, as you would expect. It's important that this model is framed in terms of molecule numbers and not concentrations, because the volume of the cell is changing. And so, but this can be done again with this assumption of balanced growth. And so this leads to two simple expressions. There's a limit where things are even simpler. There's the limit where the lifetime of the ribosomes is long, and then there we get particularly simple results. But in principle, this is we can take care of any regime. And this regime anyway will not work. For instance, if you are in a small growth condition, for instance, if you have a starved cell, you cannot work with this assumption, for instance. Okay, so I mentioned the growth laws. So the first step was to recover the growth laws from the model. So let me just remind you, for those that don't know them, what these laws are. So you have essentially two main laws that they were studied. They were discovered many years ago, but extensively studied recently by Terry Roy about 10 years ago. And in one, basically, these laws relate to how the ribosome fraction should depend on the cell growth rate. And so the first law is about this solid curve. So it tells you that the ribosome fraction should increase with the cell growth rate, and this happens because when you change the nutrient quality in the medium, even in the absence of antibiotics. And then the second law has to do with this raise here, and basically it means that when you increase the antibiotic concentration, there's an upregulation of the ribosomes. So you make more ribosomes. So the fraction is increasing, but the growth rate is decreasing. So that's what I call the second growth law. And sorry? Yeah. So in the theory work paper, they are linear, but in our calculations, we see that they are not. Or to be more precise, they become linear only in specific limits. So this is the limit that I was talking about before. But I think there's a small deviation from linearity if you look closely. So I think I don't think the dependency need to be linear. Okay, so for the model, we get a self-consistent equation, and this equation for the growth rate lambda. And so you can see this equation is cubic, and so that means that there will be a bistable behavior from this model, which was also present in the girlish work, but we have in a way slightly extended that. And so why do you have this bistable regime in some limit? So this is because you have this characteristic parameter that shows up, this lambda star, and then depending on what is the value of the pre-exposure growth rate, which is lambda 0, you have either irreversible binding of the antibiotics or reversible binding. So what this means is that in this irreversible limit, basically, it's a race between the antibiotics and the cell. And there's so much antibiotic getting in that the cell can keep up with it. And as a result, you have a kind of toggle switch. And you switch between one behavior with a high growth rate and another with a low growth rate. Instead, in a reversible limit, you have an equilibrium between the outside and the inside antibiotics. And as a result, you get a smooth dependence. So this is indeed what you see in the data. You have either a smooth dependence or a more discontinuous dependence. And you can describe many antibiotics with this, more than just these two. And they don't need to be just targeting only the ribosomes. They can be targeting other things than ribosomes, and you can still use this simple approach. So I mentioned that we were also interested in the notion of risk. So what could be the risk for a cell? So it could be the difference between life and death. So of course, all the things I told you so far, this was this was for a class of antibiotics that are known as bacteriostatic. So they don't really kill the cell; they just slow down its growth. But what is the difference between slowing down and killing? It might be that it's just how strong the antibiotics is, or at least some people are suggesting that, and our model also goes into this direction. So, because in fact, in the data that we analyzed before, one of the antibiotics is actually a bacteriocidal, this kanamycin, and so this doesn't seem to be a problem. It still has bacteriostatic effect. So my view is that there's not a strong distinction between the two, and so for that reason, you can define a form of risk or proxy for risk from the fraction of these active important molecules that are immobilized by the antibiotics. So in a reversible case, you have these dashed lines, and they turn into this solid line for the irreversible case. Okay, so another way to look at risk is to look at the half inhibitory concentration. So this is the concentration of antibiotics where the growth rate is halved. That's also a way to see these two classes of behavior, reversible versus irreversible. And then the biostability, which perhaps is the most interesting part on the story, is happening in this. Well, I'm sorry, it's not that coming out that very good. But if you are in a reversible regime, so you have the solid lines. So in this range of antibiotic concentration, you jump between two growth rates. So that's the theoretical predictions, but I think there's also experimental proof of that in this work. So you see clearly that there is a kind of bistable region here in this work for also from Terry Waugh from 2013. So I think there's hope that what? Sorry, yes. This is the reversible regime in which there's no jump. There's no bistability. Well, I mean, what I mean by bistable is that for this value of the antibiotic concentration, there are two possible growth rates. I'm not talking about really jumping. I'm just saying that these are two solutions. And this doesn't happen in the reversible case where everything is smooth. Okay, so just to finish with the story with antibiotics, we also looked at what happens when you have two antibiotics combine action, but they target two different autocatalytic cycles. So in principle, you might imagine to have a synergy or antagonism between these two antibiotics, but because of the coupling we choose, you can only have antagonism. And so the dose response curve look like that in this case. Okay, so let me just summarize and then move on to the last part, quickly. So we have proposed a simple model of the action of antibiotics, but it's important to realize it's really mean field approach. There's no population effect. And there's no fluctuations. So there's still a lot to be done to make contact with experiments which are typically done with the population of cells, and so I want to say that so the data we are trying to think about right now is in fact similar to the data that was presented by Nama earlier this morning. So she talked about experiments done in a control environment in a droplet, where basically there's a small population but within a droplet. And so this is the same technology actually is being done by several people in Paris, and in particular at the Pasteur Institute. And so they have basically droplets in which bacteria are growing, and these droplets are exposed to different levels of antibiotics. So this is a nice problem, but to work on, but it's hard because you have many sources of heterogeneity. So you have the fact that there are population effect, and it's a small population, and also there's growth rate heterogeneity, which may be related to the lag time heterogeneity, which was mentioned in Nama's talk. So I look forward to discussion because I think it's a hard problem, and personally I need interactions and feedback to address to work on to make progress on this. So let me just quickly say a few things about the second part, which is perhaps more related to the general topic of this workshop. So it's a problem where basically we try to analyze lineage statistics, and this actually may be a useful thing because, as I say, I mean we need the single cell statistics or the small population experiments to understand really what's going on with the antibiotics in real experiments. So this work was basically about trying to analyze population experiments in which some lineage may stop because, for instance, the cells could die. So I'll show you a situation where there is antibiotics again and cells die, and as a result, the lineage ends. And you might want to know what you can do with this, statistically speaking. I mean, when as I argue that this is in principle a problem to have a lineage that ends before, but there might be interesting effect associated to that. So if you're not familiar with this, I probably need to explain that there is actually a procedure that was proposed by Edo Kussel and Takashi Nozoe some years ago, and this procedure was based on two ways to sample a lineage. So one way is to sample them in the forward way, and in the other one is the backward way. So the backward way essentially means that you take a snapshot of the population and you go back in some sense along the lineage. So this is the easiest one. This is where most the experiments are most easily done. But the problem is that you may have a bias effect if you sample according to this backward sampling. And so there's for instance, here is a simple illustration of this kind of bias that you can get. If you have, in a population view, you have overrepresentation of small cells, and you see that clearly in this distribution with respect to the forward distribution. And the forward distribution or the forward sampling corresponds to what is being done in mother machine. So it's important to understand this difference, this biasing effect, in order to fully exploit the lineage statistics. You can use this difference between the two ensembles to define a measure of fitness, and I will come back to that. It's an interesting way to measure fitness from a statistical properties of lineage. So some years ago, we were interested in the question of inferring the population growth rate from mother machine data. So Ariel presented this topic, and I have to say that when we were interested in this in 2020, we had very naive view, and I think I really want to say that it's because of your help and the discussion that we had that really I understood the subtleties of this problem. I mean, because I had completely missed how difficult this is. And I'm very happy to hear that now I think you have nailed it and that you have a good control of the error in this kind of inference. So just to quickly to say a few words about this. So this question of incomplete lineage. So this shows up when there's cell death. This shows up in this setup because you lose cells, and so what the problem is that when the lineage are incomplete, is that you cannot put any weight on this incomplete lineage because there's nothing there essentially. So that's a problem in the way. In the method introduced by Kussel and Takashi, and so you need to revise that. And one way to do that is to introduce a new distribution of the forward sampling, but conditioned on survival. And so, in principle, this is the correct distribution, and from that you can infer fitness landscape more correctly, and you can also quantify the survival bias that comes from ignoring essentially this dead lineage. So we tested that with data from antibiotics, and so that's why I thought it's kind of fitting to talk about two experiments that have to do with antibiotics. So you see here the time where the drug is exposed, and as a result, you see the lineage that become extinct progressively, and as the population of cells is also going down. In this paper, the conclusion, so this was way before all these ideas about lineage analysis were introduced. So the paper concluded that there was no correlation between pre-exposure growth rate and survival. And so we wanted to check this, this statement using a more careful way to analyze lineage, I would say. And so the answer, of course, depends on which variable you're looking at and or which variable you're using in order to define this fitness landscape. And for instance, if you use as variable this pre-exposure growth rate, then you see that the three measures basically of fitness landscape both are flat. And that means that there is no, that confirms that there is no correlation. But if you look at different variable like the final cell size, then it's a different story because whether you take correctly into account the incomplete lineage or not, you may have the impression that there is some correlation that exists, which actually just coming from this this incomplete lineage. So just to to finish, the method can also be used for inference, not just of fitness landscape, but essentially inference of simple parameters that you want to know about, namely the division rate and the death rate. And so we tested this idea on this numerically in a model where you see there's a division rate and there's a death rate, which both depend on the trait S, which can fluctuate in time. And so we use these two distributions to infer R and gamma. And here, this is a test that we infer the correct law for R and gamma from the lineage analysis. So this is true. This is here we're using a simple Ornstein-Beck process, but in principle, this could work. This could work beyond. So that's it. I think I just want to finish by saying that together with Uli, we are organizing a workshop in a year from now in Munich. So the topic is a bit different, but you might also be interested in chemical reaction networks in connection with design principles for synthetic life. And I want to also acknowledge the people who are helping us in who have actually been doing this work. So in particular, the work on antibiotics is done by Barnabé, but inspired by a discussion with Charles Barrault at the Pasteur Institute. And the cell lineage work has been done with Arthur and with the help of Takashi Nozoe and Locability. Thank you very much.